The overall goal of this procedure is to image biological assemblies in liquid in order to observe their dynamic behaviors at nanometer resolution. This is accomplished by first preparing the silicon nitride microchips with an affinity capture surface to enable macromolecules to be tethered to the microchip surface, preventing diffusion in liquid. The second step is to load biological samples onto the microchip in order to recruit rotavirus particles. Next, the microchip containing the virus particles is washed with a contrast agent and then loaded onto the microfluidic specimen holder. The final step is to load the top chip and faceplate to form a closed vessel encasing the liquid specimen. Ultimately, images are recorded in the TEM to reveal features of the virus sample within the liquid microchamber.
The main advantage of this technique over other existing methods to examine entities in liquid is that our specimen do not rapidly diffuse over great distances while imaging due to the affinity capture coating applied to the microchips.
The implications of this technique extend towards real-time assessments of nanoparticle-based drug delivery systems or vaccine development against viral pathogens, because we can see for the first time how dynamic nanomachines change in solution.
Though this method can provide insight into visualizing biological macromolecules, it can also be applied to other systems, such as electrochemistry applications.
A visual demonstration of this method is critical since preparing affinity capture devices and loading the microfluidic specimen holder may present a problem to the novice user.
To begin, clean the silicon nitride E-chips by incubating them in 15 milliliters of acetone for 2 minutes, followed by 15 milliliters of methanol.